Unit 3 Cell Structure and Function. Biology is the study of living organisms. The detailed description of their form and appearance only brought out their diversity. It is the cell theory that emphasizes the unity underlying these diversity of forms, that is the cellular organization of all life forms. A description of cell structure and cell growth by division is given in the chapter comprising this unit. Cell theory also created a sense of mystery around living phenomena, that is physiological and behavioral process. This mystery was the requirement of the integrity of cellular organization for living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral process, one can take a physiochemical approach and use cell-free system to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by analysis of living tissue for elements and compounds. It will tell us what types of organic compounds are present in living organism. In that next stage, one can ask the question, what are these compounds doing inside a cell, and in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is the molecular basis of all physiological processes? It can also explain the abnormal process that occur during any diseased condition. This physiochemical approach to study an underlying living organism is called reductionist biology. The concept and technique of physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules is provided. Scientist G.N. Ramchandran, born in 1922 and died in 2001. G.N. Ramchandran, an outstanding figure in the field of protein structure, was the founder of Madras School of Conformational Analysis of Biopolymer. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformation of protein through the use of the Ramchandran plot rank among the most outstanding contribution in structural biology. He was born on October 8 in 1922 in a small town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had considerable influence in shaping Ramchandran's interest in mathematics. After completing his school years, Ramchandran graduated in 1942 as the top ranking student in the BSc Honours Physics course of the University of Madras. He received a PhD from Cambridge University in 1949. While at Cambridge, Ramchandran met Linus Pauling and was deeply influenced by his publication on models of the alpha helix and beta sheet structure that directed his attention to solving the structure of collagen. He passed away at the age of 78 on 7 April 2001. Chapter 8, Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life. The cell is the cell in all living organism. All organism are composed of cells. Some are composed of single cell and are unicellular organism, while others like us composed of many cells are called multicellular organism. 8.1, what is a cell? Unicellular organisms are capable of first independent existence and second performing the essential function of life. Anything less than a complete structure of cell does not ensure independent living. Hence cell is the fundamental structure and functional unit of all living organism. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. Anton von Leeuwenhoek first saw and described live cells. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. 8.2 Cell theory. In 1838, Malthus Skeldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissue of the plant at about the same time. Theodore Swan in 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his study on plant tissue, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Swan proposed the hypothesis that the body of animals and plants are composed of cells and products of cell. Skeldon and Swan together formulated the cell theory. This theory, however, did not explain to how new cells were formed. Rudolf Virchow in 1855 first explained that cells divide and new cells are formed from pre-existing cells, that is called as omnicellular e cellula. He modified the hypothesis of Skeldon and Swan to give the cell theory a final shape. Cell theory as understood today is: first, all living organisms are composed of cells and product of cells. Second, all cells arise from pre-existing cells. 8.3 An overview of cells. You have earlier observed cell in an onion peel and or human chick cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human chick have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material, DNA. Cells that have a membrane bound nuclei are called eukaryotic, whereas cells that lack a membrane bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cells. The cytoplasm is the main arena of cellular activity in both the plant and animal cells. Various chemical reactions occur in it to keep the cells in the living stage. Beside the nucleus, the eukaryotic cells have other membrane-bound distinct structure called organelles like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane bound organelle found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelle chloroplast in plants and mitochondria and on rough ER, that is endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome, which help in cell division. Cells differ greatly in size, shape, and activities, that is given in figure 8.1. For example, mycoplasma, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich among multicellular organisms. Human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shapes. They may be disc-like, polygonal, columnar, cuboid, thread-like structure, or even irregular. The shape of the cells may vary with the function they perform. 8.4 Prokaryotic cell. The prokaryotic cells are represented by bacteria, blue grain algae, mycoplasma, and Pipiello pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells, that is given in figure 8.2. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum, spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotic exhibit a wide variety of shapes and functions. All prokaryotic have a cell wall surrounding the cell membrane except in mycoplasma, the fluid matrix filling the cell. In the cytoplasm, there is no well-defined nucleus. The genetic material is basically necked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have a small circular DNA outside the genomic DNA. These small DNA are called plasmids. Plasmid DNA confers certain unique phenotypic character to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation. With foreign DNA, nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cell except for ribosome. Prokaryotes have something unique in the form of inclusion. A specialized differentiated form of cell membrane called mesosome is a characteristic of prokaryotes. They are essentially enfolding of cell membrane. 8.41 Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelope and the manner in which they respond to the staining procedure developed by Gram. That is, those that take up the Gram stain are called Gram positive and the others do not are called Gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interact with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cell. They also help in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called the chromatophores, which contain pigment. Bacterial cells may be motile or non-motile. If motile, they have a thin filamentous extension from their cell wall called flagella. Bacteria so arrange in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside flagella, pili and fimbriae are also surface structure of the bacteria but do not play a role in motility. The pili are elongated tubular structure made of a special protein. The fimbriae also small blister-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rock in stems and also to the host tissue. 8.42 Ribosome and inclusion body. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 50 nanometer to by 20 nanometer in size and are made of two subunits, that is 50S and 30S units, which when present together form 70S prokaryotic ribosome. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome and polysome. The ribosome of a polysome translates the mRNA into protein. Inclusion body. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion body. These are not bound by any membrane system and lie free in the cytoplasm, that is phosphate granules, cyanophyscian granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryote cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryote cell possesses an organized nucleus with a nuclear envelope. In addition, eukaryote cells have a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosome as the chromosome. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell wall, plastids, and a large central vacuole, which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all plant cells. That is given in figure 8.3. Let us now look at individual cells organelles to understand their structure and function. 8.51 Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950. Meanwhile, chemical study on the cell membrane, especially in human red blood cells, enabled the scientists to reduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. In addition to phospholipid membranes also contain cholesterol. Later biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane protein can be classified as integral and peripheral. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model that is given in figure 8.4. According to this, the quasi-fluid nature of lipid enable lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intercellular junction, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecule across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called as passive transport. Neutral solids may also move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example: sodium and potassium pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives the shape to the cells and protects the cell wall from mechanical damage and infection, it also helps in cell to cell interaction and provide barrier to undesirable macromolecule. Algae have a cell wall made of cellulose, gelatin, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and protein. The cell wall of a young plant shell, the primary wall, is capable of growth which gradually diminishes as the cell mature, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which hold or glues not the different neighboring cells together. The cell wall and middle lamella may be traversed by a plasmodesmata which connect the cytoplasm of neighboring cell. 8.53 Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosomes, and vacuole. Since the function of the mitochondria, chloroplast, and peroxisomes are not coordinated with the above component, these are not considered as a part of the endomembrane system. 8.531 The endoplasmic reticulum. Electron microscope study of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structure scattered in the cytoplasm that is called the endoplasmic reticulum, that is given in figure 8.5. Hence endoplasmic reticulum, the intracellular space into two distinct compartments, that is luminal inside endoplasmic reticulum and extraluminal cytoplasm compartment. The ER often show ribosome attached to their surface. The endoplasmic reticulum bearing ribosome on their surface is called rough endoplasmic reticulum, RER. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum, that is SER. Rough endoplasmic reticulum is frequently observed in the cell actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipids. Lipid in animal cell, lipid like steroid hormones are synthesized in smooth endoplasmic reticulum. 8.532. Golgi apparatus. Camilo Golgi first observed densely stent reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sac cisternae of 0.5 micrometer to 1 micrometer diameter, that is given in figure 8.6. These are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging material to be delivered either to the intracellular target or is secreted outside the cell. Materials to be packaged in the form of vesicles form the ER, fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remain in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus because they are released from its trans phase. Golgi apparatus is the important site of information of glycoprotein and glycolipid. 8.533 Lysosomes. These are membrane-bound vesicle structure formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all type of hydrolytic enzymes, hydrolases, lipases, proteases, carbo, carb, carbohydrates is optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. 8.534 Vacuoles. The vacuole is the membrane-bound species found in the cytoplasm. It contains water, sap, excretory product and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuoles than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist, food vacuoles are formed by the engulfing of the food material. 8.54 Mitochondria, unless specifically stand, are not easily visible under the microscope. The number of mitochondria per cell is variably depending on the physiological activity of the cell. In terms of shape and size, also considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter 0.2 to 1 micrometer, average is 0.5 micrometer, and length is 1 to 4.1 micrometer. Each mitochondrion is double membrane-bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae towards the matrix, that is given in figure 8.7. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouse of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecules, ribosome 70S, and the compartments required for synthesis of protein. The mitochondria divide by fission. 8.55 Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plant. Based on the type of pigment, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essentially for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophyll, and others are present. These gives the part of the plant a yellow, orange, or red color. The leucoplast are the colorless plastids of various shape and size with stored nutrients. Amyloplast store carbohydrate starch, example potato. Elyplast store oils and fats, whereas the aleuroplast store protein. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaf. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelle, having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell to the Calamnodomonas, a green alga, to 20 to 40 per cell in the mesophyll like mitochondria. The chromoplasts are also double membrane bound. Of the two, inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma, that is given in figure 8.8. Thylakoids are arranged in stacks like the piles of coins called grana or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamella connecting the thylakoids of the different grana. The membrane of the thylakoid encloses a space called the lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecule and ribosome. Chlorophyll pigments are present in the thylakoid. The ribosomes of the chloroplast are small are smaller are smaller are smaller 70s than the cytoplasmic ribosome, that is 80s. 8.56 Ribosomes are the granular structure first observed under the electron microscope as dense particle by George Pallet in 1953. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane. The eukaryotic ribosome are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller subunit, that is given in figure 8.9. The two subunits of 80s ribosomes are 60s and 40s, while that of 70s ribosomes are 50s and 30s. Here as is Swedberg's unit stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. 8.57 Cytoskeleton, an elaborate network of filamentous proteinous structure consisting of microtubules, microfilaments, and intermediate filament present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in each cells are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. 8.58 Cilia and flagella. Cilia and flagella are hair-like outgrowth of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that one of the eukaryotic flagella. The electron microscopic study of cilium or the flagellum so that they are covered with plasma membrane. Their core, called the axonema, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as the 9 plus 2 array, that is given in figure 8.10. The central tubules are connected by a bridges and is called enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal body. 8.59 Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structure called centrioles. They are surrounded by amorphous pericentriolar material. Both the centrioles in a centrosome like perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles from the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cell. 8.510 Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin. By Fleming, the interphase nucleus of a cell, when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli, that is given in figure 8.11. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membrane with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosome on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membrane. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect the names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, example erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix or the nucleoplasm contains the nucleolus and chromatin. The nucleoli are spherical structure present in the nucleoplasm. The content of the nucleolus is continuous with the reset of the nucleoplasm, as it is not a membrane bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called the chromatin, but during different stages of cell division, cells so structured chromosome in place of the nucleus. Chromatin contain DNA and some basic protein called histone, some non-histone protein, and Also, RNA, a single human cell has approximately 2-meter long thread of DNA distributed among its 46 23 pairs chromosome. You will study the detail of DNA packaging in the form of chromosome in class 12th. Every chromosome visible only in dividing cells essentially has a primary constriction or the centromere on the sides of each of which disc-shaped structure called kinetochores are present. Centromeres hold two chromatids of a chromosome. Based on the position of the centromere, the chromosome can be classified into four types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constriction at a constant location. This gives the appearance of a small fragment called the satellite. 8.511 Microbodies. Many membrane bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cell. Summary: all organisms are made of cells or aggregates of cells, vary in their shape and size and activities function based on the presence or absence of a membrane bound nucleus and other organelle cells, and hence organism can be named as eukaryote or prokaryotic. A typical eukaryotic cells consist of a cell membrane, nucleus, and cytoplasm. Plant cells have a cell wall. Outside the cell membrane, the plasma membrane is selectively permeable and facilitate transport of several molecules. The endomembrane system includes Golgi complex, lysosome, and vacuoles. All the cell organelles perform different but specific functions. Centrosome and centrioles form the basal body of cilia and flagella that facilitate locomotion. In animal cells, centrioles also form spindle apparatus during cell division. Nucleus contain nucleoli and the chromatin network. It not only control the activity of the organelle but also play a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. They are of two types, rough and smooth. Helps in the transport of substance, synthesis of proteins, lipoprotein and glycogen. The Golgi body is a membranous organelle composed of flattened sac. The secretion of cells are packed in them and transported from the cell. Lysosomes are single membrane structure containing enzymes for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with the mitochondria, help in the oxidative phosphorylation and generation of adenosine triphosphate. They are bound by a double membrane. The outer membrane is smooth and inner one folded into several cristaplastids are pigment containing organelles found in plant cells only. In plant cells, chloroplasts are responsible for trapping light energy, essentially for photosynthesis. The grana in the plastid is the site of light reaction and the stroma of dark reaction. The green colored plastids are chloroplasts which contain chlorophyll, whereas the other colored plastid are chromoplasts which may contain pigments like carotene and xanthophyll. The nucleus is enclosed by a nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, the cell is the structural and functional unit of life. Thank you friends for listening this audio, and please post your comment if you want any kind of improvement. So मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए बाय एंड stay tuned with biology critic and enjoy the knowledge. Okay, bye.